So we're here on behalf of SV Industry interviewing Bart deplancke. He works in the SV school, and we're going to learn a little bit more about him. You can go ahead and introduce yourself.
Well, the name was already said. I'm Bart deplancke. I'm a professor here at the School of Life Sciences, also a member of the Institute of Bioengineering. I've been at the EPFL since 2007, so I'm a full professor here at the EPFL. And I'm also Vice Dean of Innovation, which is probably also one of the reasons why you're here. In terms of research, what we're doing in the laboratory is, as some of you know, we're actually very much interested in understanding the function of the genome. So the series of nucleotides that are present in each of our cells. And so how does that genome operate? How does it actually make a cell the way it is? And how do these cells together assemble themselves into an organism? And so to study that, we've sort of separated or partitioned our lab in 3 different pillars. One of them is actually sort of focused on understanding more metabolic phenotypic variation, obesity, and so forth. So understanding how the genome and variation in that genome actually leads to these sort of metabolic phenotypes. Another is actually focused more on more fundamental mechanisms of how genes are expressed and how they are differentially expressed between individuals, depending on genotype. And in order to study that, we also have a 3rd pillar in the lab, which basically focuses on developing technologies to address these questions. And so some of the technologies that we're a little bit known for, I guess, is specifically tailored towards analyzing systems at the single cell level. And so we have quite a bit of technologies under development, but also software trying to actually now look at systems at the resolution of a single cell.
Yeah. So your company your lab is involved in a lot of industry related topics. So you also developed a few companies in your throughout your career. So could you
That's correct. That's correct. Yeah. So
tell us a bit about that. I don't know, company starting creation, like how it goes from like a lab idea to a company.
Sure. I mean, it's often organic. It's not necessarily planned that we will make a company. I mean, projects basically sort of develop a little bit organically, and all of a sudden there's an opportunity. So to maybe make an example, the first company I was involved in was a company that actually focused on data management, especially in sort of the changing environment of biology, which is basically rendering it more data science. Yeah. And, what we realized basically together also with colleagues and friends sort of together discussing this is that most of the data management systems basically were not adapted to this changing environment where basically biology started to acquire more and more of that type of data. And so basically there was an opportunity there to do better and to actually make sure that these data management systems actually would be adapted to actually, for example, welcoming sequencing data, lots of reads, high volumes and so forth. So we sort of got our heads together and said what needs to be done. And from there, basically, we started developing a particular system. And this was also not, of course, only me, but actually also friends outside life sciences, which was actually quite interesting. Because, for example, one of them came from the telecom business, where he was laughing with us because he says, when you describe big data in life sciences, we call that small data in telecom, right? So the systems that they had developed in order to, to sort of track all the data trafficking, basically, in the telecom business actually turned out to be very efficient and useful also for life sciences. So that's where the opportunity came. And then the second company we recently launched basically, again, sort of grew organically because we developed a new technology, a sort of workflow to analyze the expression of genes in bulk samples, which made the entire workflow actually much cheaper, but also more amenable to higher throughput. And so the dream of many pharma companies would be to perform drug screens and not only look at what happens to those cells, but also to receive digital information, namely what happens to the genes that are expressed in those cells when we expose these cells to drugs, right? But doing this sort of, for thousands and thousands of compounds and each time creating an RNA-seq library is still way too expensive. And so with our workflow, we're actually really trying to sort of bridge that gap and sort of propose, for example, pharma companies a solution now that, would allow them actually in a sort of reasonable cost add this sort of a digital component to their sort of workflow of drug development and design. And so here we didn't really initially have this in mind. We actually sort of turned the question upside down because we were working on single cell technologies. And then we said, but actually some of these principles could also be used for bulk analyses, which was quite interesting. So we sort of said, okay, let's develop this. And we published a paper, and then we sort of got bombarded by requests from all over the world, basically say, oh, but can you help us implementing this workflow? This is great. I mean, this is, both industry and labs. And then basically the people who had developed this in the lab, we said, well, actually this idea of maybe actually using this workflow and actually making it in something more automated and accessible for all researchers, independent of whether you're from industry and academia. They were very intrigued by this idea. And so sooner or later they said, well, actually we had maybe in mind to continue in academia, but now that there's opportunities there, why don't we launch a company based on this new workflow and see where we can get? And so that's sort of, I think, often the way it goes. You don't really have something in mind initially, but then the opportunity presents itself and you launch yourself into it and see where you can go.
Okay. So this second company is alithea.
Alithea Genomics. Yeah, correct. Yes, exactly.
Okay.
Yeah.
So is there anything like you're working on now that you'd be interested in talking about? I don't know.
Well, yeah, I think, we also have another project in mind that we probably will translate into a company. And this is actually to do with technology that we're building basically that will allow us to sort of fairly uniquely not only analyze which genes are expressed in the cell, but to immediately link that also to sort of the phenotype of that cell. And why is that important? I mean, sort of the big dream here is basically that ultimately, if you know each time basically the image of a cell, and then you have the transcriptome, so you know actually what this cell is because you have the ground truth, so to speak. You know based on transcriptomics, you can sort of then say, okay, well, this is a cancer cell maybe coming from liver or whatever, right? So just to give you an example, the dream is that maybe actually if you get enough of that data, that ultimately it may be sufficient to actually, for example, do a blood analysis and see certain cells pass by your microscope and say, through machine learning, for example, but I know this cell. I've seen it before, and I know what it is because I had the ground truth before.
So some like supervised learning model.
Exactly. Okay. Yeah. Well, it could be unsupervised because sometimes we don't know actually what the model will find out that actually distinguishes this cell from another. Okay. But it's sufficient for us to be able to classify those cells, And the technology that we're building will allow us to do this pairing of modalities, mainly the transcriptome and the image. And I think we can think of many examples of where this could be useful. One of them is diagnostics, but also to learn, for example, how certain cellular features, organelles, lipid droplets, whatever, how they arise and what the molecular determinants of these phenotypes are. So, and I already have sort of the people who develop this technology, they again have in mind, we really want to launch a company on this. So we'll see. In 1 or 2 years, there might be a third company on the list here, but we'll see.
So you spent a lot of your career at UPFL creating companies. How is the relationship between like professors and the industry? Like, is that something that's very fluid, or?
I mean, it's sort of a very flexible ecosystem here, and I think we have to give credit to the EPFL to creating an environment where professors feel compelled to actually sort of support their students in creating these companies. First of all, because we sort of beyond our 100% appointment in principle, we have 20% additionally that we can sort of dedicate to this kind of endeavors. Right. So in principle, 1 day a week, we could actually also sort of put time into the company, which allows, especially in the beginning, a lot of support that we can give because, as you may know, of course, it's very critical when you sort of wean yourself off the EPFL and become really an independent company where you have your own sort of buildings and your own lab space or whatever. So during that period, I think it's really important that the PI can sort of keep on guiding. And at the same time, also the people from the company can sort of keep on using and benefiting from the EPFL ecosystem, right? And so I have the feeling that basically the EPFL is really amenable to that. And so they understand these issues and they try to actually really support us in that way.
So, and I was wondering, this is kind of a personal question, but well, for me, how does intellectual property manage? Because if you do research under the hood of a BFL, it's like it belongs to them in theory. So,
yes, absolutely. And I think that's a win-win situation. I think it would be wrong to try to sort of sneak IP out of the EPFL and sort of use it to your own benefit. Because there's no question that the EPFL name is also a big sort of label for the startup company. So it's sort of a win-win. And so, for Alithea Genomics, for example, we actually did file IP. And the idea there is basically that we simply then license that in an exclusive license from DPFL, right? And of course, the conditions under which this happens need to be negotiated with DPFL. Yeah. But again, I feel like DPFL is accommodative because they also understand the strain that startups are under. I mean, I think we are all interested for our startups to be successful. So having overly demanding requirements, would not be useful for anybody, because then the startup may not be attractive to outside investors, and then DPFL would have nothing at the end of the day. So again, I think here the ecosystem understands this fully, and I think everybody is willing to play that game because, like you say, this workflow was actually developed in the lab, so it would be really sort of wrong to actually try to take that out and then sort of pretend as if this didn't happen. So it's a good relationship.
And so your career has been like very intertwined with industry. Have you ever been tempted to take the leap and go over to industry?
Yeah, there's been the occasional emails and calls of of interest, but I have to say, basically, what I really appreciate in sort of having interacted and still interact with many people from industry itself, I really sort of really like the idea of having this intellectual flexibility of doing what I think is cool, right? Is interesting. And then still actually having the luxury in a way to see if some of these ideas which came to fruition in the lab, whether they also have relevance in society. And I think that's actually sort of a bridge that you can seldom make in industry, because you don't necessarily have the intellectual flexibility. I mean, you're typically sort of already put into a pipeline that is underway, and you have to sort of follow the way. And I also like a lot of fundamental research, right? And having to give that up in order to make that bridge to industry, I think I would start to miss it a lot. Because yes, it's tempting. I may make more money at the end of the day, but I think I've over time I've really realized this is not what makes you happy. It makes you happy to get up and say, "Oh, wouldn't it be great if we can study this?" Or,
ask cool questions.
Ask cool questions. And I think that's actually what sort of allows me to stay here in academia, but at the same time, I'm always intrigued by seeing how far we can push this in terms of societal relevance.
Of course. So have you seen any different trends in how the industry has evolved, and how the relationship between like schools and industry have evolved, or academia and industry?
Yeah, I think over time, I think there has been a period, I think, maybe I'm sort of exaggerating a little bit, but there has been a period where basically many companies try to do a lot in-house. And I think now we're sort of in a period where basically a lot of companies realize the potential of campuses, universities, to actually do lots of the groundwork actually over there. So there's these industry contracts that they say, okay, we don't really know where this will lead, but we'll give you a little bit of money and we'll supervise this a little bit from far, and then let us know what actually comes out of that. And I think that's useful, because they know that there's sort of an intellectual ecosystem here that basically is very fruitful, very multidisciplinary, which in companies you don't always have. And so that's a little bit what they're trying to benefit from. That said, what is now very interesting in sort of the last years almost is that industry is starting to attract a lot of talent from academia, especially sort of in data, rich domains. And I've been sort of surprised by actually sort of moves from some of the leaders in the field, for example, the single cell field, like Aviv Regev, for example, who's really considered to be a world leader in single cell biology, omic technologies, and so forth, who has been recruited by Genentech, part of Roche, in order to sort of lead the research developments there. And I think why, I don't know, you have to ask her, of course, but I think, and I've seen this from other sort of leading figures, I think why they are attracted to go to industry is because the industry has unlimited resources almost, especially compared to academia. And so since biology is becoming very data intensive, it's often a little bit of a frustration in academia that we cannot generate the amounts of data you need in order to, for example, do really proper machine learning, but you can do it in industry. And at the same time, what is really interesting, they tell these world leaders like Aviv, they say, come to industry, help us understand these problems, and we will generate the data for you. And at the same time, we also allow you to still play around a little bit with your
Okay.
fundamental research.
So there's an understanding on the industry side that people still want to have the liberty of
Absolutely, because I just explained that.
Yeah.
For me, this would also be a requirement. This is, of course, not all, rare that you would get this. But now we see this as well to Roche, where they now created this new organoid center, right? I mean, they were able to recruit a colleague of ours, Matthias Luthoff, over there. Again, sort of under the same understanding.
Yeah.
There's a little bit of an academic component to what they're trying to do. And at the same time, they give so many resources that I think he can now do things that you can only dream of.
Yeah.
So there's very interesting developments. And I'm sort of keeping my eye on those as well.
Okay.
Yeah.
Okay. So as you mentioned before, you're also Vice Dean for Innovation here at EPFL. So we're also here to talk about Catalyze for Life and what that is. So could you explain a little bit?
Sure, yeah. So Catalyze for Life sort of started from the realization that the innovation potential of the life sciences school or any kind of life sciences activity doesn't have to be necessarily the school itself, but there's also lots of life sciences going on in the other schools in a way, was not fully tapped, right? And specifically in 2016, we had an audit by an external committee. And they said, how is it possible that you are a Swiss Federal Institute of Technology, whereas basically the IP patents, is somewhat lacking a little bit. Now, of course, the School of Life Sciences, as you know, is still one of the youngest schools here on the campus, right? And so it was sort of natural that we first sort of tried to actually, make sure that our foundations are solid, that the academic spirit is there, and that students get what they need in terms of actually sort of courses and biological engineering inspired materials. But I think it was time to actually take this comment a little bit more seriously. And so we said, okay, what can we do about this? And so from there, basically, Catalyze for Life was born, with the idea of actually trying to sort of have people in place that can really go talk to PIs and often students, because the PI might not even be aware of what might be possible. And some students in the labs might say, but I actually want to maybe create a company of this. And not all PIs are industry minded. And they're like, well, give it a try. And then those students will say, but how can I do this? And so we're there to help them, right? So we're now actually sort of a team of people who have this type of experience. And we try to support sort of little projects that would allow to demonstrate, for example, to an outsider that indeed this project that they had in mind has potential. For example, it's on the verge of creating IP, necessary IP, and the PI says, yeah, but I'm not necessarily interested, or I am interested, but I don't have the funds. Well, we try to give them a little bit of funds in order to at least get a proof of concept, such that they can go a little bit wider, right?
So could you bring it, like, in the steps of startup creation, could you tell us when and how Catalyze for Life actually helps? And
so typically the researchers are still very much in the lab, and they're sitting on this interesting data. They may have filed already a patent, or they're thinking about it, and they're sort of lacking maybe some final experiment that would really be crucial. And then they knock on our door, because we really are not in the stage where the company would already be incorporated. When that happens, we're sort of out of the game, Catalyze for Life. We're really there for the very early beginnings of what could be ultimately a company, or maybe a very solid patents that could then be licensed and so forth, that sort of would demonstrate our innovation within the school, or sort of anything life sciences.
So more something like, as soon as you have a quick idea, you think something of it,
Well, the idea is not sufficient. I always say ideas are cheap. Actually demonstrating that they're valuable is something completely different.
Sure.
And so the idea already has to be worked out to a certain extent, and they have to sort of convince us that all they need is sort of maybe one more key experiment, or maybe something like that, and then we're happy to support them. And so that's now becoming something quite recurrent. So we've actually sort of already supported, I think now almost 18 or so projects, which are now at various stages. Some have led to startups, some have led to really interesting patents that are now licensed out. And so we're actually fairly happy with those developments. But at the same time, we also realized basically that, it's not only about the research, it's also actually exposing students in general, basically, to the needs of industry, and realizing that, and thanks to actually our current director Gautam Matra, who actually really comes from industry and has years and years of experience in that, we sort of set up a new course in sort of exposing students to the needs, for example, for regulations. What does it take to file a patent? What does it take to bring a drug to the market, or a biotech instrument to the market, and so forth? And this has so far been a 6-week course, which has been incredibly well liked, apparently, because the feedback we always ask is very positive. And what really actually motivated us altogether is the type of feedback where students are saying, well, I put this on my CV, and when I interviewed, I really realized that is why I was sitting there, right? It was really a distinguisher, from others who applied for the same job. And they said, but you already showed interest in the industry and you already know a little bit about it. So this was really a sort of a game changer. But we also realized that now, given that we run this from the end of August till the end of September, roughly, that actually most of you guys would not have access to it, because it's so far been mostly PhD students and postdocs. And so now we're actually in the works of creating sort of a master's course.
Okay.
That could then be available to all the master students here on campus. And I think I'm really looking forward to that endeavor, because I think it probably will attract quite a bit of attention.
Of course.
Yeah. Mm-hmm.
So could you maybe go through an example of a company that we might have heard of from Catalyze for Life? Or
I was thinking of one example that we just funded, actually, just to give you an idea, because it's sort of now interesting to see where this goes. For example, there's Elisa Rocchio's group in Israel, who actually has a very interesting project working on catepsin inhibitors. And the ideas of these catepsins is that they're sort of proteases. They break down proteins. And they have sort of been implicated, especially in metastasis, because the idea is that they break down the extracellular matrix, which makes the tumor cells more mobile, right? Because they're sort of no longer constrained by this matrix. And so it's already been sort of thought for a while that designing or finding inhibitors of these catepsins could sort of slow metastasis, right? And there have been clinical trials on this, and they were unsuccessful. And we don't really know why exactly, but one of the ideas is that they were too non-specific, sort of broadly available, but not specific enough to actually target the tumor cells. Okay. And what Elisa's lab now has done is to actually couple those inhibitors or sort of to targeting antibodies, for example, to target lymphoma, B-cell malignancies. And she's demonstrated already, has some nice proof of concepts where basically, this looks very promising. Okay. And so basically we just funded now basically that sort of more thorough research project to, for example, optimize the bonding between the antibody and the inhibitor, also provide some in vivo validation of sort of what was already seen in vitro, to then really sort of bring this entire file up to a level where maybe it will attract investors or they will start a company. That's at least the idea, right? But there's also been, for example, a company that was recently launched by Mickey De Palma, actually, that also actually works on for example, we supported them early on, where they actually try to modulate sort of immune cells and make them more active again within the tumor. And they have some very intriguing technologies and know-how actually of how to do that. And this has attracted actually already outside investment. So I know there's sort of some nice figures going into the company, and they've actually now started the company, starting all the way actually from the Catalyze for Life investment. It's not that this was crucial, but it was a nice part of it. See what I mean? So these are sort of two examples.
Of course.
Exactly. Yes. Yeah.
Okay. Yeah. I think we kind of wrapped up the questions I had prepared. But if you have anything you want to add, anything you want to advertise,
up to you guys. No. I think
we're good.
I think we're good. We have 23 minutes of recording. So.
Okay, so we can, and so this is going to end up in clips.
Sure. Yeah, I understand. Okay, I tend to talk a lot.
That's a good thing. So
that's nice.
So maybe a quick outro. Thanks for watching, and I'll see you next interview.
Thank you.